Hello, my name is Marko Bajc, and I will be talking about the cost of forest genetic monitoring. Now, successful implementation of forest genetic monitoring will depend on many different factors, one of them being securing the long-term funding for this activity. This means that researchers will undoubtedly have to answer the question to policymakers and funding agencies, what is the cost of forest genetic monitoring? With that in mind, costs of all activities for forest genetic monitoring were logged throughout the duration of the Lifegenmon project by all participating partners. In the end, as accurate as possible, an assessment of cost was performed, and the purpose of this is to analyze the structure of total cost of forest genetic monitoring and to identify approaches for potential reduction of costs. Before we dive into the cost assessment itself, a basic information about the forest genetic monitoring system developed in the Lifegenmon project will be presented. This monitoring system is based basically on three indicators, namely selection, genetic variation, gene flow and mating system. Each of these indicators comes with their own set of parameters to be monitored, which fall into two different types, verifiers and background information. Additionally, we have devised three different levels of genetic monitoring: basic, standard, and advanced. The number of parameters that are monitored for each of these levels increases from the basic through standard to the advanced level. And with that, the amount of information that we gather increases, but also do the costs. Now, chapter 2 will be focused on the cost assessment itself. Before we look at the numbers, we are going to have a look at what different activities and cost categories were considered in cost assessment calculations. First, of course, we are interested in what's total cost of different levels. The same goes for different indicators and verifiers, of which there are many. Different activities. There are 5 basic activities that need to be performed during genetic monitoring, namely plot selection, plot establishment, field observations, sampling, and laboratory analysis. We're also interested in, are there any differences in total cost of monitoring for the 2 analyzed species? And are there any differences between countries that participated in the project? And finally, what is the structure of total cost per different cost categories: materials, effective work, and traveling? In order to have cost assessment as comparable and representative between different countries, we needed to take certain considerations into account when doing the calculations. For one, all the costs were evaluated for an average 10-year monitoring period. Both of the evaluated species are stand-forming, and we expect that costs for scattered species are going to be higher. Costs are presented for optimized activities, as performed by fully trained and experienced personnel. All costs include value-added tax, traveling distance to plot, or actually distance to the plot, was set to 100 kilometers for both species and all 3 countries, in order to avoid having differences related to different traveling distances. The same applies to the number of visits to the plots that are necessary for field observations of field parameters. As for materials, cost of different materials and cost of labor, actual costs as reported by participating partners were used in the calculations. So first, we are going to have a look at total cost of forest genetic monitoring per different monitoring activities. There are 5 different activities: plot selection, plot establishment, field observations, sampling, and laboratory analysis. Values presented here are for both species and all 3 levels and all 3 countries. One thing that needs to be further explained is that for the Greek values, we have actually 2 entries. The reason for that is that in Greece, Field observations were primarily performed by photographic observations, which resulted in significantly higher costs as compared to visual observations as done in Slovenia and Germany. So in order to have directly comparable cost assessment, we also calculated the values for total cost for Greek monitoring activities as if they were performed by visual means only. Certain things become evident very quickly when we look at this chart, and that is that the cost of genetic monitoring increases substantially from one level to another. Second, there is a very clear major contributor to total cost in terms of different activities, field observations. When we look at the same values just in a relative form, so looking at the percentage of total cost per different activities, we can clearly see that field observations are the dominant contributor across all levels, species, and countries. Next is the breakdown of costs per cost categories. Here we have three categories: materials, which include all consumables and outsourcing necessary to perform all of the genetic monitoring activities. Next is effective work. Here it's the cost of man-hours necessary to perform different activities, but this category does not include hours, man-hours spent traveling. These are included in the third category, the traveling, which besides the hours traveling includes also mileage, subsistence, and other traveling related costs. If we look across levels, different levels and species, we can see that effective work is basically consistently a significant contributor, but traveling can also contribute significantly to total cost. Now, since the cost of traveling depends on the distance to the plot, we also evaluated the effects of traveling distance on the total cost of forest genetic monitoring. This was done only for European beech in Slovenia, but the same goes also for other countries and species. So besides the standard 100-kilometer distance to the plot, we also considered two other distances to the plot in the calculations: 25 kilometers and 175 kilometers. And we can see that the effects of distance are very, very significant, pronounced. If we compare total costs for a 175-kilometer distance to the plot, we can see the total costs almost double compared to 25-kilometer distance to the plot. And again, at longer distances, traveling costs account for more than 50% of total cost of genetic monitoring, clearly identifying that traveling costs are, on the one hand, very variable contributor to total cost of genetic monitoring, and can, at longer distances, become the major contributor. So this is a factor that needs to be considered when we are planning on starting genetic monitoring. Next in line is assessment of total cost of forest genetic monitoring per indicator. Here the results are fairly predictable. We can see that selection, the indicator selection, remains the major contributing factor to total cost across all levels and all species and countries included. The reason for that is that field observations, which have already been identified as the major contributing activity to the total cost of monitoring, are included in the indicator selection. If we look at relative contributions of different verifiers and background information to the total cost, we have to first consider that there are different parameters and different total number of parameters monitored for different levels of monitoring. So if we start at the basic level, we can see that there is a very consistent structure of costs across all three countries. Again, we're looking only at values for beach, but for all three participating countries. And here verifies natural abundance, flowering, and fructifications represent the three major contributing parameters to total cost. The same three parameters remain significant contributors at standard level, but are overtaken by two additional parameters, namely bud break and senescence, which become the two major contributing factors to total cost on standard and advanced levels. If we look at contributions of individual parameters that are based on molecular genetic analysis, we can see that individual contributions are fairly minor, and altogether they represent a moderate contribution to total cost at the standard level, and similar is observed at the advanced level, where the contribution is slightly higher and approaches up to 25% of total cost. One of the things we also did with our assessment is that we calculated an average total cost of forest genetic monitoring per monitoring level. Here, values from all three countries and both species were considered in calculating average values. And we have two tables here. One is focusing on the monitoring activities, and the other on different cost categories. All values are represented also with standard deviations. And if we look at total, we can see that for basic level, we expect the total cost on average to be around 7,300 euros, for standard level 26,000 euros, and for the advanced level 76,000 euros. Again, field observations are highlighted as major contributor in terms of activities, and effective work and traveling as major contributors in terms of different cost categories. However, traveling will depend largely on the distance to the plot. This brings us to our final chapter of this presentation, conclusions and recommendations. Now, if we look at different levels of genetic monitoring, we have already established what average cost of monitoring per level are. And if we look at the ratio, so how the cost increases, we can see that we can expect approximately 3.5-fold increase of cost from basic to standard level, and an additional 3-fold increase from standard to advanced. In terms of activities, field observations are clearly the dominant contributor across all levels and species, and represent on average 88% of total cost for the basic level, 75% for standard, and 62% for advanced level. In cost categories, we can expect that effective work is generally going to be the dominant cost category, but traveling can contribute significantly, and at longer distances for traveling to the plot can actually become itself the major contributing factor to the total cost. When we analyze the differences between species, we found no significant differences in total cost of monitoring on the basic level, but this changes at standard and advanced levels. And generally, the total cost of genetic monitoring of silver or King Boris's fir is lower than that of European beech. That is for 16% standard level and 28% lower at advanced level. The reason for that lies that for the ABS, we are not monitoring one of the parameters, the senescence. And similar differences are also expected for other stand-forming broadleaf versus conifer comparisons. However, for other types of species, such as scattered species and species that hybridize and/or form clones, we expect that the total cost of monitoring will be higher. in part due to additional fieldwork and also additional laboratory analysis, which for hybridizing and vegetatively reproducing species will also have to be done at the basic level. Comparing the countries, the highest cost of forest genetic monitoring is recorded for Germany. If we look at average values, we see that the costs in Germany are 85% higher than that for Greece and 73% for Slovenia. Now, the most significant contributor to this difference is a much, much higher cost of man-hour for all staff categories in Germany compared to Greece or Slovenia. And all other contributing factors to this difference are much, much less significant, almost insignificant. Is there a way to reduce cost of genetic monitoring? Certainly, there are some recommendations that we can make to keep the costs tamed as much as possible. First, it is recommended that you always use fully trained and experienced personnel to carry out all genetic monitoring activities, because trained and experienced personnel will perform their jobs faster and with fewer mistakes, saving time and money. Organize your work well. This goes for field observations and laboratory work. In terms of field observations, certain activities, certain measurements can be done during the same visit to the plot. So plan your work to do them during the same visit instead of traveling to the plot on several separate occasions. In terms of laboratory analysis, don't analyze every batch of samples that comes into the laboratory. Wait until a sufficient number of samples are delivered and then analyze them in bulk. This will utilize the maximum capacity of your machines and will reduce cost per sample. It is recommended not to use photographic documentation of phenology as a routine procedure. As we have seen on the example of the Greek approach, photographic approach increased the cost, total cost of monitoring for standard and advanced levels by over 100%. Perhaps an unexpected solution to cutting down costs is consider international collaboration. Many neighboring countries share the same species and environmental zones, so they don't really always need to conduct genetic monitoring for each of these species and environmental zones separately. Traveling is one of those aspects of cost assessment that has become perhaps the most interesting and surprising. It is, of course, the most variable cost category and can contribute significantly, as shown before. So in order to reduce the contribution of traveling cost, following measures can be taken. During the plot selection phase of the monitoring, if we find several different plots that meet all the necessary criteria for a forest genetic monitoring plot, choose the one that is the closest to the institute. Secondly, get Forest Service or equivalent organization in your country involved in forest genetic monitoring to perform field observations and measurements. Local foresters will have to travel shorter distances compared to personnel of research institutions on average, thereby cutting down traveling costs. Also, when performing time-consuming field work, such as plot establishment, consider to have your field personnel stay overnight locally and not drive back to the institute. In such a way, more effective work can be done per day and fewer trips necessary to complete the task. And this is particularly advisable for plots requiring long traveling times. And finally, if possible, allocate more personnel for labor-intensive fieldwork. So instead of having just 2 people drive in a car to the plot and do the work, and having to return the next day to complete the task, try and find and send 4 people to the plot. So again, more effective work will be done per day, and traveling costs per person will go down. This concludes my presentation. I thank you for your attention.